就可以了。
Ladies and gentlemen, let's get our laptops up and settle down. I've gone for it. Sit down, sit down. I've got on, just sit down. All right, ladies and gentlemen, welcome to today's class. Let's get settled in. Kevin, have a seat, please. Right. So, welcome to CSI Dulwich. Today, you will be what we call crime scene investigators. Last lesson, we learned about food molecules, and we learned about how to test these food molecules. Now, why do we learn these skills in science? Because they are used in the real world. And I've got one of my year 13s over in the corner here who can attest to the skills that she has learned in year 10 and 11 and year 1 that she uses in that very lab there herself. So the skills that you learn actually are of use. And we link it today with something that we call CSI, or crime scene investigation. Crime scene investigation is the act of collecting evidence from a crime scene and then testing it against knowledge, against what we know, to see if we can identify a criminal. So, there has been a crime in this very lab. Over in the corner there, at 1 a.m. last night, a nefarious person came in and stole some really important data from that computer. We've got our iGen students that are using artificial intelligence, AI, to identify intestines, and it's a really, really secret process. So the data on there is really important, but it's been stolen. It's gone. Now, as crime scene investigators, it is your responsibility to help us catch the person who has committed this crime. We have managed to obtain CCTV from the cameras outside this very room, and we found somebody coming in at that time. So what we did is we identified who we thought they could be. We've got 3 suspects. We've managed to narrow it down to 3 suspects, and it's these 3 here. We've got Mr. Darling, known as the butcher, Mr. Mackay, the baker, and Mr. Greg, the candyman. One of these 3 people came in last night and stole our data, and it's up to you to tell us who it is.
Attention! Test for today is nearly closed. Please go quickly to test now. Thank you. 请注意，今日核酸检测即将结束，请尽快前往，谢谢配合。
So for the learning objectives today, written on the right-hand side over here, there are 3 learning objectives. Number 1, you need to be able to state what the tests for the food macromolecules are. You're going to need to perform these tests, and at the end of it, you're going to use a little bit of education technology to write up your report to send off to the detectives so they can make an arrest. Okay? So you've got 3 tasks today. In the first task, for the first third of the lesson, you have got a worksheet which contains the transcript of the conversations with the butcher, the baker, and the candy maker last night. And it's your job to listen, to read it, to discuss in groups what they said, and answer the questions based on the information there. The second task, you're going to then collect an unknown sample from one of our crime scene investigators in the corner over there, Miss Lynn. She's got a collection of unknown samples. Now, I actually don't know what they are. I purposely made it so I don't know the answer either. So you're going to take in pairs. So for the first task, you're going to work in your table group. When it comes to the testing, you're going to work in pairs. for you guys, you're going to work in threes, and you're going to take one sample, and you're going to test it to identify who you think is the bad guy based on the information given. And task 3, you're going to take that information and you're going to individually, singularly, write up a report based on the template that I'm going to share with you. Okay? Does everyone understand the task that has been set? Are we all good for that? Okay. So let's start off with the first one. We have 15 minutes. You are going to take the worksheet and work in groups and go through and answer the questions. So 15 minutes. Now, as we do this, you can talk to each other in Chinese or English, it's up to you. However, when it comes to writing the answers, please write them in English. Once you have finished, you can hold those notes in your notebook to help you identify the criminal at the end of the lesson. So, yep, there should be. I actually don't know what they are. They could be one, they could be another, they could be other. So please, absolutely, discuss with each other. Don't actually do it in silence. I'd like to hear you discussing and talking to each other. Yes. I can try and find one. I'm not sure. Unfortunately, Frey, I only have a pencil. However, you can draw that mistake sometimes. Okay, so let's read this. Do you want to be detective, Phil, or do you want to be the butcher? You can be the butcher. So I'm going to read this bit, you read this bit. Frey, listen. So, Mr. Butcher, where were you last night during the hours of 9 and 11 p.m.? Beef? Why beef? Oh, really? What's in protein? Huh. Can you tell me more about protein? Okay, well, thank you for your help, Mr. Butcher. So, from what you've said, is there anything that comes out that sounds not true? Is there anything in that sentence, in those sentences that the Butcher said, that don't sound right to you? What do you think? Oh, okay.
You need a protein bar. You don't know about today.
Well, what is he said there that doesn't sound correct about proteins? What do you know about proteins that is different to what the Butcher is saying?
Monosaccharides.
Have a look at your notes, Wesley. Come on, this is what you need to be doing. I'll come back to you and see what you think the answer is. So, you're already writing stuff down. Is there anything that you've noticed that you think doesn't sound quite right? Monosaccharides are not in protein. Oh, tell me more. Where are they? Glucose. Is glucose a monosaccharide? Good. Okay, good. So there we go. You found out what he got wrong. So if that's the case, if he said the monosaccharides are the monomers of proteins, but it's wrong, what's the correct answer? What are they? Yes, they are amino acids. There you go. Okay, and keep going. We'll look at the next one. Give him a hand. Let's see how the guys do. Alan, how are we getting on? Good. Yes, that is correct. That is exactly what he said. So now we've got the third question. So, gentlemen, 3 ways in which your body needs protein. Can you think of 3 ways that your body uses protein? Go on then.
Structure.
Good. Wait. Structure. What do you mean by structure?
Hair, nail.
Hair and nails. Okay. What else?
Hold bones together.
Good. Yeah, they hold our bones together. They hold our bones and muscles together. And enzymes are made of proteins. Excellent. Good. And what are enzymes important for?
Increase the speed of reaction.
They increase the speed of reactions. Very good. Well done. Good. Move on. Sorry? I did start. Time is not long. 6 minutes has gone by, right?
That's 14.
Okay, so I need to remember that actually it was set to 20 rather than set to 15. Good. Yes. These are IG1s. There we go. Fixed. Excellent. That is correct. So what do they contain? What are it?
Contains
That
oxygen.
is correct. They are composed of amino acids. We say the word composed. Move on to the next one. Have a look at the other two, because I want to try and finish this within the time frame.
然后我们看一下。
Give them a hand. Let's see how the guys get Alan, how are we getting on? Good. Yes, that is correct. That is exactly what he's saying. So now we've got the third question. So, gentlemen, 3 ways in which your body needs protein. Can you think of 3 ways that your body uses protein? Go on then.
Structure.
Good.
Logistics.
Wait. Structure. What do you mean by structure?
Hair, nail.
Hair and nails. Okay. What else?
Hold bones together.
Good. Yeah, they hold our bones together. They hold our bones and muscles together. And enzymes are made of proteins. Excellent. Good. And what are enzymes important for?
Increase the speed of reaction.
They increase the speed of reactions. Very good. Well done. Good. Move on. Sorry? I did start. Time is not long. 6 minutes has gone by, right?
That's 14.
Okay, so I need to remember that actually it was set to 20 rather than set to 15. Good. Yes. These are IG1s. There we go. Fixed. Okay. Excellent. That is correct. So what do they contain? What are they made of? Fantastic. That is a great answer. They are composed of amino acids, which means that's what they're made of. When we say the word composed, we mean that they are made of amino acids. Good job. Move on to the next one. Have a look at the other two, because we want to try and finish this within the time frame. There you go. So we're getting on. What are we on to now? Ooh, the baker. So what is the baker? What do you think the baker is going to be involved with? What monosaccharide? What chemical? What chemical have we learned about so far that might be involved in baking? Yeah, carbohydrates. What give me an example of a carbohydrate you find in bread? Great, good. So, in the transcript, the baker has made an incorrect statement. Do you know what it is? Very good. Where is glycogen found? Yes, you. Yeah, you and me, we, us, glycogen. So, starch is what you find in plants, whereas glycogen is what you find in animals. That's the big difference there. Okay. You could have also said cellulose, but technically bread might contain a bit of cellulose because it does come from plant cells, and plant cells have cellulose. So yes, definitely glycogen. Good, well done.
它一个很硬的 structure， 然后呢，这个面包里它大量含这个化合物，含 starch， 对吧？那 starch 它是由什么组成的？是 glycogen 跟 amylopectin。
What's wrong? What's up? Yes, the 3 different polysaccharides. So there's these 3 here. Which one would the baker be? What would? So he said cellulose and glycogen. Which of those might be correct? Why? Yeah, exactly. It's found in animal. It's not found in animals. Sorry, glycogen is found in animals, but not in plants. I mean, technically, you could get glycogen into bread, but you would accidentally need to have a mouse in the bread as well. No, that's not. That totally never happened.
Bread is not the most tasty.
Potatoes, absolutely, are potatoes. But are they found in proteins? So remember, we learnt about 3 macromolecules: carbohydrates, proteins, and fats and lipids. Mm-hmm. Monosaccharides, are they found in carbohydrates, or are they found in proteins, or are they found in fats and lipids? They're found in carbohydrates, exactly. So there's your see there. Your monomer of protein, amino acids. Okay, so you've got proteins. The monomer is amino acids. It's saying the monomer of carbohydrate is actually monosaccharides. See here? So you've got your notes here that say that carbohydrates are made up of monosaccharides, but you've got your notes here that say that proteins are made up of amino acids. See? Got it? Good. Fantastic. It's okay. Nothing I need to do. Well, what's flour from? What's flour made from? Yeah, from. Exactly. Well, I mean, when you're grinding up flour, you're grinding up cells. So there will be cellulose in bread, of course. There you go. If you had animals like bugs and stuff, you're not going to oh, they know this. Alright, ladies and gentlemen, I'm going to give you 2 more minutes to work on this. If you haven't managed to finish all 3 of them at lunch, when you eat lunch, you're going to have excess amounts of sugar from the food that you eat. Where's most of that sugar going to go? I mean, you're going to use some of it for energy, but where's the rest of it going to go? Some of it might be converted into fat if you have too much sugar, but what else will it put it into? What's the storage? Remember you just talked about this earlier. What's the storage molecule of sugars? Is that in humans? It's not in humans. Glycogen. So your body will convert the sugars into glycogen and fats. That's what happens to these sugars. All right, ladies and gentlemen, let's stop there for a moment. Can you please put your pens and pencils down and look towards me whilst I talk for the next one? So the next task is we're going to be doing some lab testing. You're going to have 20 minutes to do this, which should be enough time because we've looked at this, so we already have this skill, so it's about refining them already. So before we start, this is what you're going to set it up like. You have this device called a spotting tile, and with the pipettes that I've given you, you're going to place a small amount in the spotting tile and add the test to them to observe if a color change is going to happen, okay? Rather than using test tubes. But there are 3 test tubes available, which you're going to have to use for the Benedict's test. I'm already boiling the water at the back of the room there, so I can help get the water to a temperature that's going to make it work quite quickly. All right, and I'll come along and switch it on. Let's have a look at what we already know. So this is burettes. We'll put starch, protein, and glucose into each well. So we'll put a bit of starch into that one, a bit of protein into that one, a bit of glucose into that one, and we'll add burettes to each one. Which one are you expecting to change colour with burettes? Is it starch, protein, or glucose? Frey, what do you think? So burettes, what does that test for? Starch, protein, or glucose? Okay, it's for protein. What colour did we say burettes turns? I'll let you think about it. I'll come back to you, Fred. You think about it, and I'll come and ask you in a second. All right, Lily. Same thing again. Starch, protein, glucose, but this time we're adding iodine. Which one do you think will react with iodine? What color change? Dark blue. Okay, Fred. Protein. Did you get it? Think of another color. Begins with the letter P. Like protein. Good. Yes. So protein turns purple. And iodine turns a bluey black color. We're also going to be doing the Benedict's test, and it's going to be set up like this. Starch, protein, glucose. Which of those 3 do you think will change color then, Alan? Which one? So when we do the Benedict's test tests for starch, protein, or glucose. Avery, do you want to give him a hand? Benedict's. Burette's tests for protein. Benedict's tests for glucose. What color did we say would change? What color did it change when we test for glucose? Orange. Yeah, it turns an orangey, reddy color. So this is what we should expect as our standards. You're going to create a control, but underneath you're going to be testing unknown. So in a second, when you get ready, I want you to go to my assistant, Detective Lynn, over there and collect an unknown sample. You're going to place the unknown sample in 2 more wells, add the burettes and iodine to see if they test for that, and you're going to have another test tube on your desk to test for Benedict's, use Benedict's test to test the unknown solution as well. Okay, so set them up like that. You have got 20 minutes in which to complete this task. On you go. You're going to work in pairs in this test. And on the back paper, here, on the back page, on the last page, is where you're going to record your results to help you with your answers. Okay? Good. There we go. Carry on. When you do this experiment, make sure you have got your seats under and that you're standing up so you don't so you're not accidentally going to be unsafe. Make sure you wear safety goggles as well to protect your eyes, and if you have long hair, have it tied back.
叫太小了，因为一手型配，拍到啥也看不见，都带着不行。
I've got some boiled water. I'll bring you some boiled water to make sure it works. Yeah, you're saying it is boiled already, so it shouldn't take too long to heat. I'll come along and start the fire in a minute.
蹦出来了，是的。
There we go. Remember, Mark, you must be standing when you do this. Will you chair over?
要换起的泡沫了。你先把这个滴到这里。
需要点火吗？
发现了。
Oh, well done. Can I have the clicker? I'll help the boys over there. Or you don't know?
可以，还得弄吧。我还没什么啊。
There you go. Okay, good. Right, gentlemen, you've got two unknown solutions? Yep.
No.
Yes, you do. Unknown 2, and you've got your unknown other number. Did you collect your unknown samples yet?
哦，你第一台吗？
One for
这个第一台吗？第三个红了，需要吗？闻到，因为它要两个 unknown。
你可了解上面两个是吧？上面那个是
你们的 unknown 借我们第一下。
专门的一瓶 unknown 那个，
对，
不是一样的吗？
这不是 unknown 吗？哎，那也是 unknown。
对，一样的呀。
一样的，为什么不一样？他们要两种 unknown 呀。我。
就是你们两两一起，是让你们两两。
对，就是
什么两瓶？
所以大家要记住 unknown。
你闻的是一个人一瓶，两个人一瓶啊？
对，两个人一瓶。
是不是有没有问题？
不是。
什么不是？两个人一瓶。
这里原皮的，原皮是这样，皮也不行。哇，
啊，本来生八水就是好的。啊，然后分别方式有点小鬼，这个这个是空的是吧？去把那个试剂拧开来，然后这个管滴进去，然后随便点，用到手上来了，不会有事吧？
啊，你没有自己的手好。
然后
别这是 unknown。
Yeah. Can you please go get some safety goggles, or at least put your safety goggles on?
Can I wear my glasses?
You can, but you need a different set of safety goggles.
Yes.
These ones are safety glasses. These ones are regular glasses.
拿走，这 unknown 上电子。
Just curiously, did I hear?
还好。
Yeah, so I'm gonna get your test tube set up for the Benedict's test. Now, because even though you guys have your own unknown samples, you can still use the same starch, glucose, Benedict, starch, glucose, protein, salmon controls. Here we go. What's in these ones? Unknown starch glucose. We're not testing protein. Because you're already on protein. Oh, so what do we think it is? What's the unknown sample? Glucose. So, from our three wanted men, the butcher, the baker, and the candy maker, who do you think is most likely to be the bad guy? Why the candy man? Because the candy man. Sorry? What's candy made of? Exactly. Candy makes sugar. That's why we eat candy, because it's so tasty. What does meat do? Sweet. Yeah. So, like, if it was the butcher, which macromolecule would the butcher be associated with? Protein. What about the baker? Starch. And then the candy maker is something that's do glucose. This one is starch. Okay, perfect. So you're all back. Yeah, I want you to so your report's going to be different. Which one? Why is that black? Interesting. So what's this one testing for?
Oh, starch and benedict.
This is starch. Wait, what's in here?
I just,
Okay, yeah,
I just.
no.
No wonder it's an interesting color.
It is an interesting. Right. Listen very carefully, gentlemen. I will say this only once. Starch goes into one of these. That protein goes into another one. Glucose goes into another one. Yes? So you've got your three controls. You've got two more test tubes. One for your unknown sample and one for your unknown sample. The 5 of them then go into the beaker to see if there are any color changes. And it's only Benedict's test that we add to the test tubes. Okay? Only Benedict. However, your results look interesting. What's happened here?
So this is a varied test. The first one is starch, second one is protein, the third one is glucose,
And the fourth one?
fourth one is unknown.
Unknown sample. So what can you conclude from here already from your results?
So due to the color change, our unknown.
Maybe protein. Good. Well, but the only way you're going to find out for definite is by completing the iodine test and the Benedict's test to make sure that it's only protein. And from that, you should be able to identify who the bad guy is. What would protein be associated with? Meat. So who do you think the bad guy is? The butcher. Good. Excellent. Keep going, guys. Let's do this one quickly because this one's going to take time. Ladies and gentlemen, time check. You have 11 minutes. Can you take
Benedict's 的话是
How are we getting up? Hey, be sure back hands. Which one are you? So, what was in here? What was in this one? Unknown. What was in this one? What was in this one? Protein. Now this is a very interesting result. Let's have a look at this. So if we take this one out. So here we have a reaction, even though it only contains starch. Now, why might that happen? Why have we got a slight amount of glucose here? What could possibly cause it? What's starch made of? Starch made of carbohydrates. Now, starch is made up of sugar molecules that are connected together. Now, it shouldn't react with Benedict. However, two things could have caused this change in color. Number one, human error. So when you are adding the starch to it, maybe you got a little bit of glucose in the solution, maybe the solution has a bit of glucose in it. Or another thing that could happen, the heat was enough to cause the starch to break down and split into its glucose molecules. Hence why it's really. But we can write this one off as it's an accident. Your real color change is shown here. As you can see, the differences are quite immense. You've got your red coloration to show that this one contains glucose. You've got this one that's not changing any color at all, which is likely your protein, your starch, and what was this one? Glucose. So you can see from the results that the solution must be glucose. Okay. Right, so you've finished all your reactions so far. Write down in here what the color changes were, and then that way you should be able to identify who the bad guy is. Is it the butcher, the baker, or the candy maker? 8 minutes, ladies and gentlemen. How are you going to go, gentlemen? It's just one coming around. Where's your lever? Oh, so which one was the unknown sound? Oh, yeah, okay. Doesn't matter, the water's going to be sun down there, so it's degrees.
哇，够多呀！如果这里面挤的，那我往哪挤啊？没有那个肺疗这个里面没有肺液不能挤回去。这三个桶。不是啊，水不够了。你看到吗？那个够了。
Is the unknown sound?
要去选择方向了，不是。
我没了。
我没了。
我们的那我提前我弄到那个地方没有多少。
How about your other results?
哪个去拿了？
红点。
Good. So who do you think about?
可以。
哦，哇，你还是太老了，没有肺啊。
Sorry, say again.
先说两个先。
All right, tidy up. We have got 6 minutes until we start the next one. So use this time. Don't touch this until it's falling down. Otherwise you need to Hey Greg, how are things? Have we got our results? Yeah. Excellent. That's right. Into our page. So you have something to Once you have done that, can you please tidy up? Now, this one might be a bit hot, so just leave it for the moment. But this these things Right, 15 seconds. Let's just sit down again, please. Sit down. Great, Stephen. I appreciate the cleaning, but please have a seat now. Excellent. All right, so the last part of today is the actual product that you're going to produce. You've had a read of the suspects' transcripts. You've tested the results, and you should hopefully have a firm identification of who you think the perpetrator is. So, quick question. Hands up if your group discovered the unknown solution to be protein. Alan. Anyone else? Protein? Was there only one group with protein?
Oh,
Protein, good. Now, let me ask you this, gentlemen. Which bad guy is associated with protein? Would it be the butcher, the baker, or the candy maker?
Butcher.
Why the butcher?
Because meat is from
Very good, because meat contains protein. Fantastic. Hands up if you managed to identify starch as the unknown solution. Lily's group, Irene's group, Yolanda's group. Sylvia, who do you think is the bad guy then? The baker. Why the baker? Talks about bread, and what's bread famous for containing? Starch, exactly, very good. And finally, who chose glucose? Who identified glucose? Olivia's group and Kevin's group. Okay, Ava. So if it's not the butcher or the baker, it must be the candy man. Why the candy man? Because candy contains sugar, contains glucose. Brilliant. Right, ladies and gentlemen, thank you very much. Now, the last thing I want you to do, and this is your sort of exit ticket, this is the final report, what I want you to do. On your computers, can you access our group Padlet? And I want you to use this structure when it comes to writing your answer. So, according to the evidence, the criminal is who your criminal is. The evidence I have to prove this is, so write down the evidence that you have obtained to prove this. Now, I did also ask to include a photo, but I never actually asked you to take photos. So if you don't have photos, that's okay. All I want is your written report. If you have taken photos, fantastic. You can add them to your report. The QR code is actually based on the bottom of the piece of paper that I've given to you as well. So if you want to type it out, it's there. If you want the QR code, it's there. No, it's just like 1, 2, 3, 4, however many pieces of evidence you have. What I will do is I will share our group Padlet on the board so you can see what we're doing. There we go. This is just from yesterday. Let's see. You can see what comes up. Make sure that when you are writing it, you're writing it with your name and not anonymously. If you do write it anonymously, make sure you put your name on it. In fact, just don't use it anonymously, just write your name on it. So this will take us up to the end of the lesson, which is 5 more minutes. Right answer first, this according to the evidence, the criminal is, the evidence is this. So write it actually with that part. Criminal is baker. Very good, Ava. Ava's our first report. Our unknown solution is glucose. The Benedict's turned into orange because candies contain sugar glucose, therefore the criminal is Mr. Candyman. Mr. Candy. You managed to get in, Jason? Ooh, I'd like you to do your own though, Jason. That's what I did ask for. That's part of our 21st century skills, Jason, to be able to use computers. Life is all about computers. You can't say it's inconvenient to use computers. Good job, Alan. Well done. Okay, question. Who is this person? Who is this? Yeah, name. You edit it and put your name on it so I know who you are. Thank you. Three minutes, gentlemen and ladies. 3 minutes to go. Where do you find hemoglobin? You said here hemoglobin carries oxygen. Where do you find hemoglobin? Good. Right, here's an A-level question. How many molecules of oxygen does hemoglobin? What does one molecule of hemoglobin carry? Good job, Rose. Excellent. I'll tell you what, I'll ask you that in year 12. Yes. Excellent. Well done, Terence. This looks good. We found that our unknown solution is glucose. Ah, but Terence had a comment in. Who's the bad guy? Huh? That's the whole point. You're writing this to the detectives to tell them. You're like, well, actually, Detective O, we found that the bad guy must be the Candyman because it contains glucose. Listen, ladies and gentlemen, before I dismiss you for today, let's have a look at whether or not we've gathered our learning objectives. Just a quick thumbs up, side thumbs for still need to work on it, thumbs down. How do you feel about, are you able to state the tests for glucose, for starch, and for protein? Are you able to tell me those names and how it works? Not how it works, but what the color changes. Yes? No? Still working. Still working. Good. Still working. That's okay. That means you're revising. How confident do you feel about being able to perform a test? If you have to do an experiment in a couple of weeks' time, testing for these things again, how confident do you feel that you'll be able to do it again? Yes, no, maybe. Congratulations. Okay, good. All right, Mark, pay attention. And finally, how confident do you feel about using our Padlet tech for being able to write reviews and reports? Good man. All right. If you haven't finished, get it finished. If you have finished, take your lab coats off, put them away, and thank you very much for taking part in today's lesson. I appreciate your attendance, smiles, cooperation, collaboration, and general joy. All of you.